Hello all. Today we'll be discussing important questions in food and nutrition. Now, not all important questions, we'll be discussing one particular pattern. Okay. Mostly, which is seen repeatedly in the previous years. This type of questions has come repeatedly. So we'll be seeing what type of questions are there. Mainly, we'll be just focusing on match the following questions. Okay. Now, this is the type of question. I hope it is visible. It's not very clear, I know, but I have just taken a photograph from a previous year question paper. You can see this. I just wanted to show you that it's been repeatedly coming in different questions like 2019, 2016 papers. These were seen, and you see these two papers are from two different years. Okay, so here you see Ninhydrin test, Benedict's test. See, you see, both of them are very familiar, very similar in nature. But these are two questions, and which has come in very close years. One was in 2016 and one was in 2018. You see, this is a pattern which has been repeatedly seen. And another type of question. So these are different type of questions which are repeatedly seen. Okay, so the basic is, see, the first question is, match the laboratory test given in list 1 with their chemical compound in list 2. Okay, you have seen Ninhydrin test, Benedict's test, Geldal, iodine, etc. And you will see what the test is meant for. Okay. Some of you might be familiar with these tests. Like I'm sure all of you, if you have studied in your BSc classes in food and nutrition, your practical classes, you must have done the Benedict's test or the 2,6-dichlorophenol indophenol test for vitamin C's, etc. So some of these tests might be very familiar and some of them might not be familiar. Okay, so we'll be discussing these tests today. So you see, all these are previous year questions. Okay, so so this is a common type of questions that have been repeatedly asked, and it's not directly given in the syllabus that tests for these things are asked, but the questions have been repeatedly asked. Okay, so let's see. Carbohydrates, we all know what carbohydrates are, and basically the tests for carbohydrates are usually they are chemical procedures used to identify and characterize different types of carbohydrates, including monosaccharides, disaccharides, polysaccharides. I'm sure all of you must have known that carbohydrates, there are different types of carbohydrates, monosaccharides like glucose, fructose and all, disaccharides like sucrose, lactose and all. And there are polysaccharides like starch, cellulose, glycogen, etc. Now these tests, the tests that we do in laboratories are meant for distinguishing which type of carbohydrate it is. And also sometimes there are certain tests like the very basic test that we did in school days, like this test for starch. For example, you must have done it in your school days. That is, on a piece of potato, you add some iodine drops and then the color changes to blue-black. So iodine can change starch to blue-black. So you see that there is a presence of carbohydrate or there is a presence of starch. Now there are two types of tests basically. Whenever we do any test, it's like qualitative test and quantitative test. Qualitative means it's not about testing the quality. It is basically if this thing is there or not. For example, the test I mentioned, that is the iodine test, is a qualitative test. You don't know how much of starch is present, but you know if it is present or absent. Okay, that is what is a qualitative test. It is just to determine if there is a particular compound is there or not, if it is present or absent. That is what is qualitative. And quantitative is how much is present. Okay, for example, there are certain tests for proteins like Millen's test, etc., wherein you add one reagent to a certain thing that contains protein. For example, egg white, and the color changes. So you understand that there is protein, but you don't know how much protein is there. To understand how much protein is there, you need to do some quantitative tests. So we'll be discussing about qualitative, quantitative tests for carbohydrates, proteins, fats, etc. We'll not be doing to a lot of detail, but we'll be discussing the names, the procedure, and also what it gives. So the first test for carbohydrate is Molish's test. Please remember these names, because usually you will get the names and you have to match. You need to know. Okay. So this is like, it's a very general test. Molish's test is a very general qualitative test to detect the presence of all types of carbohydrates. So any carbohydrate, if you any type of things, like be it a potato, a sugar, a fruit, anything, if you just added this Molish's reagent, it will give you a violet or a purple color due to the formation of a complex compound. So a positive result is indicated by a violet or a purple color. So you see this, there is a violet ring that you can see, a reddish purple ring at the junction of two liquids. So you have the solution wherein you have the sugar solution and you add the Molish's reagent and you get a purple or a violet ring. Okay. So the benefit of this test is it is possible for all types of carbohydrate. So you cannot differentiate the type of carbohydrate. It gives positive results for monosaccharides, disaccharides, reducing sugars, not reducing sugar, etc. Okay. Just the presence or absence of a carbohydrate. So that is Molish's test. Please make note of it, Molish. The reagent is Molish's reagent. The next is Benedict's test. This is Benedict. You might be familiar with the Benedict's reagents. Excuse me. The Benedict's test. Now, this is particularly to detect the presence of reducing sugars. Okay. Now, what do you mean by reducing sugars? There are reducing sugars and non-reducing sugars. Reducing sugars are that sugars that can reduce other compounds and undergo oxidation reaction. Okay, they undergo, just simply put, they have a free aldehyde or a ketone group. Now we know how the carbohydrate structure is. Carbohydrate structure is either they have a aldehyde group, like glucose, for example, they have a aldehyde group. It's a hexose sugar. Okay. Whereas fructose has a ketone group. So if it has a free aldehyde or a ketone group, then it can undergo oxidation because the group, the aldehyde or the ketone group is free to undergo oxidation reaction. Whereas if there is no free aldehyde or ketone group, they cannot undergo oxidation reactions. So they cannot reduce other compounds. That is called as non-reducing sugars. Now the example of reducing sugar is glucose, fructose, maltose, lactose. And non-reducing sugars are sucrose. Now the reactivity, they can, so reducing sugar means it will have certain, that is, there are different tests too. Mainly 3 tests are there to distinguish between reducing and non-reducing sugar. Please make note, reducing sugars will give a positive Benedict's test, Fehling test, and Tollens test. 3 tests. Okay, we'll be discussing these tests in detail. So first is the Benedict's test. So here you say it is a detect the presence of reducing sugars like glucose, fructose, etc. So here you are using copper sulfate in an alkaline solution. It will react with reducing sugars to form a reddish brown precipitate. A positive result is indicated by a color change from blue. So you know that the Benedict reagent is blue in color. Copper sulfate, we know it is blue in color. So copper sulfate, the Benedict's reagent is also blue in color. So when you are adding the Benedict's reagent to any solution which contains carbohydrate, what will happen if there is a reducing sugar is present, then there will be a color change. If it is a solution of sugar is a non-reducing, or sucrose is a non-reducing sugar. So what happens is the table sugar that we use every day, it is non-reducing. It will not give a positive Benedict test. It will give a negative Benedict test. Okay. So here you see, you're taking a Benedict's reagent, add some carbohydrate solution to it. You can boil over a flame and then let it rest. And you see there is a positive reaction. Now this is a semi-quantitative test. You mentioned, see the semi-quantitative. Why? You will know how much, the color change. The color form depends on the amount of reducing sugar present. For example, you see this is the clear solution of Benedict's reagent. And when you add the carbohydrate solution, if it is not much, there is only a small amount of reducing sugar is there. The color change will be only green in color. If it is a little more, it will be orange in color. And if it is high, then it is around a brick red color. Okay. So that is why it is called a semi-quantitative. Depending on the amount of reducing sugar present, the color change will vary. Now coming to the next test is Barford. Barford, B-A-R-F-O-E-D. Barford's test. Now this is a test to distinguish between monosaccharides and disaccharides. Benedict's test was to differentiate between reducing and non-reducing sugar. Here it is monosaccharide and disaccharides. So this Barford reagent, they will only react with monosaccharides like glucose. Okay. And galactose. These are all monosaccharides to form a brick red precipitate. Now this is also here, you can see there is a precipitate formation. So you see, Barford is for distinguishing between monosaccharides and disaccharides. Next is Felling's test. Felling's test is also very similar to Benedict's test. This is also used to detect reducing sugar. Here also, it is reddish brown precipitate, just like Benedict's test. It is very similar. The next test is Celimano's test. Celimano's test, it is also called as resorcinol test. Here it gives a, when there are the hexoses. Hexoses means there are fructose is a fructose contain keto group, whereas glucose contain a aldehyde group. Now ketohexoses, keto means ketone. It's like fructose. It is a specifically for ketones. There is ketohexoses and aldohexoses are there. Okay. Celimano's test is for ketohexoses like fructose. So fructose on treatment with hydrochloric acid will form 5-hydroxymethyl furfuryl, which on condensation with some compound called as resorcinol gives a cherry red complex. Okay. So this Celimano's test is usually done for to identify if it is a ketohexose or a aldohexose. So glucose will give a negative reaction. Okay, glucose will not change color to red. Okay. The next is Ozazone test. Now this is usually conducted as a confirmatory test. After, for example, you are given a test tube containing a solution, an unknown solution. First you do Molish's test, then you can do Benedict's test, Felling's test, Celimano's test, etc. So after these series of tests, you understand if it is is it a monosaccharide, is it a disaccharide, is it a ketohexose, it is an aldohexose, etc. And the last test is, for example, in the Molish's reagent, if you will only know if it is a carbohydrate or not. And in the Benedict's test, if it is reducing sugar. The Celimano's test, you will know if it is it has a ketohexose. Suppose you get a positive test, positive for all these. Now the last test is ozone test. Now this will identify and differentiate specific type of carbohydrates, okay, especially for reducing sugars. Now this is a reaction of reducing sugars with something called as phenylhydrazine in the presence of an acid, and it forms crystals called as ozone. Please make note of this ozone, which has characteristic shapes. Okay, so where are these shapes formed? You can see this after the reaction, when you if the form of crystallization happens, and then you look at the crystal inside a microscope. That is, you mount the sample on a microscope and you can see these type of images. And depending on the shape of the crystal, you will know what type of carbohydrate it is. You see these fine long needle shapes, sunflower shapes or flower shaped, it's maltose, and fructose is needle shaped. No, different shapes are there. Depending on the shape of it, you will know. So this test is called as ozone test. Now, tolerance test is to detect reducing sugars, especially aldehyde. If Selivanov test was for ketosis, tolerance test is for aldoses. Okay. So here, silver ions react and these tolerance reagent is added, and you get presence of a silver mirror on the inner surface of the test tube. You can see this. When aldehydes are absent, the tolerance test will give a negative result, and when there is a aldehydes are present, they have a dark gray precipitate or a silver mirror, which is, it forms a very glossy, shiny appearance on the walls of the test tube. Now the most simplest test, I've kept it at the last. This is the iodine test, or it is also called as Lugol's test. When you hear Lugol, don't be scared. It's just a very simple test for starch to detect the presence of starch. You just add iodine to the solution of whatever unknown solution of carbohydrate. If it turns blue, like blue, black or purple kind of type of color, you can see like this, ujala type of color, it means that there is starch molecules. A positive result is indicated by a color change to blue, black. So remember the name, it is iodine test or Lugol's test. For example, if you get the question as Lugol's test, don't be scared. Just remember it is just plain iodine test. So in a nutshell, this is the different types of tests. You see Benedict's test, it detects reducing sugars in a sample. Barford test is differentiating between monosaccharides from disaccharides, because Barford test is positive only for monosaccharides. Selivanov is differentiating ketosis from aldoses. Mollishes, it is detecting the presence of any carbohydrate. This is the most universal test, that is Mollishes test. Iodine test, presence of starch. Ozone is the most specific type of test which you can clearly identify the type of carbohydrate. Felling's test, again reducing sugars. Tollens test, again reducing sugars. Mainly aldoses. Now coming to test for proteins. The first test is called as biuret test. Biuret test is a test that detects peptide bonds in a sample. Okay, we know that proteins are made up of amino acids, and amino acids are put together in a chain, and in between amino acids there are certain bonds called as peptide bonds. Okay, now when you do these type of tests, What are you going to determine? For example, you have an unknown solution and you want to know if there is protein in it or not. So you do these qualitative tests. So what are the different qualitative tests for protein? One is biuret test, ninhydrin test, then you have Millen's test, xanthoprotic test. These are the basic tests that we all do in our labs in food and nutrition. Students will be very familiar with these because they are done in the BSc level, which you might be familiar, but you need to know these names, biuret, what they test, and if it is a qualitative or a quantitative test, etc. Okay. Biuret is a very simple or a very basic test. It's just different. For example, you get an unknown solution. For example, you have egg white. Okay. What you do is you nicely pour some water into egg white, beat it very nicely and dilute it, and you keep it as a sample. And then you are doing the biuret test. So here the test is simply sample is treated with a dilute solution of copper sulfate and sodium hydroxide. And when you see if there is a violet color, it means there is a presence of peptide bonds are there, which means there is a protein. Okay, so what is the principle? Peptide bonds will form complex ion with copper ions in the alkaline solution, which gives it a violet color. So it is a qualitative test. As I have mentioned, it just gives you an idea if there is protein in it or not. It will not give you what type of amino acid it is, what type of protein it is, etc. It will just give you an idea if there is protein in it or not. Okay, by detecting the peptide bonds. Next one is ninhydrin test. Ninhydrin test basically detects the primary amines in a sample. Okay, here you see if biuret test is a very a very basic test. Here it is used in forensics for fingerprint development and in some cases for detecting amino acids. That is, for example, when you go to a crime scene, if there is a presence of protein, for example, if it is a hair or any part of a human body or an animal, tissue will contain these amines. Okay. So that is why it is used in forensic science for fingerprint development, etc. And also for detecting amino acids. So here the compound is ninhydrin. Okay. Ninhydrin, so what happened is the sample, whatever sample you have, it might be any sample, it will be treated with a solution of ninhydrin and there is a development of blue or purple color. It means there is presence of primary amines. So ninhydrin reacts with these primary amines to form a deep blue or purple colored compound. Okay. So ninhydrin is a test for determining if there is primary amines or not. Next comes Millen's test, which detects phenolic compounds, including mainly tyrosine. Okay, so Millen's reagent is there, just mercuric nitrate dissolved in nitric acid. It reacts with the phenolic compounds to produce a red solution. So Millen's test, if you are adding Millen's solution or Millen's reagent to a sample, development of a red color indicates the presence of phenolic compounds. So this is a qualitative test for specific amino acids like tyrosine. If tyrosine is present, you will get red color when you add Millen's reagent. Next is the xanthoprotic test. Xanthoprotic test is for detection of aromatic amino acids, particularly tryptophan. Aromatic means it has a ring structure or a benzene ring is there. So xanthoprotic test is very specific for aromatic. Okay, aliphatic or the chain structured amino acids will be negative for this test. Now here, the sample is treated with nitric acid. Development of a yellow color indicates the presence of aromatic amino acid. You just see that, when you handle these nitric acid in a lab, if just nitric acid falls on your skin, just on your skin, if a little bit of acid falls, it becomes yellow. Okay, because of this reaction. Because of it reacts with these aromatic amino acids which is present in our body. Okay. So nitric acid reacts with aromatic compounds to form nitro derivatives. So these here xanthoprotic test, remember when you are writing your notes, write it as xanthoprotic test, aromatic amino acids. Okay. And what are aromatic acids? Which are ring-shaped structures. For example, tryptophan. Next is quantitative methods. Okay. Now, how do you measure quantitatively? There are a lot of methods now. Now there are like methods very specifically you can understand how much of protein is there by methods like, HPL, high performance liquid chromatography, gas chromatography. By, those are very advanced methods. But for a very simple method or in a laboratory setting, no, HPLC, all those machineries cost lakhs of rupees. Now, in a lab setting, you can't, in a college or a school, you can't afford to have such a huge machinery. But you can do gel dal, which is also pretty expensive. If you're doing a gel dal apparatus, it is a little complicated. However, it is cheaper than the HPLC method. Now, here you're not determining how much of protein is there, but you're determining how much of nitrogen content is there. Okay. Now, proteins here, the samples are digested with concentrated sulfuric acid. When you digest the sample, it converts the nitrogen to ammonium sulfate, and the ammonia is released and distilled and titrated. Okay. And here, you can understand from the amount of nitrogen present, you can calculate the protein present in it. Because the amount of nitrogen in any food material is very directly related to the protein content. So you can see that when you are, there is a nitrogen to protein conversion factor. Okay, there is the amount of, for example, after this gel dal method, you get the amount of only nitrogen content. Okay, when you multiply that into 6.25, that is called as the nitrogen to protein conversion factor. Now this factor will give you the amount of protein. When you get the amount of nitrogen, you multiply it into 6.25 and you get how much protein is there in the sample. So sample is first digested with concentrated sulfuric acid. Then the ammonium sulfate is produced, and then the ammonia is released and distilled and titrated, and you get how much of nitrogen is there. From that, you will get the amount of protein. Here also, Dumas method is also almost like the same. There is sample here is also combusted. Nitrogen gas, it is burnt, and nitrogen gas is measured directly using a gas chromatograph. Okay, so this is an easier method compared to gel dal method. Easier in the sense it's quicker, because gel dal method, here the protein digestion itself takes a lot of time. So in order to compensate for that, the Dumas method, it directly burns the sample, is combusted, and directly nitrogen gas is released, and that is measured using a gas chromatograph. Okay. So here also the amount of nitrogen is then multiplied into 6.25. Please make note, nitrogen to protein conversion factor is 6.25. The amount of nitrogen into 6.25 will give you the amount of protein. Now let's go to, so these are the basic tests for proteins. We have just 4 of them. Gel dal, I've mentioned 4 basic qualitative and 2 quantitative. Biuret, ninhydrin, Millin's test, xanthoprotic test, and qualitative is gel dal and Dumas. Now coming to test for fats and oils. Now here the main, I am not going to very much details because I've given details about carbohydrate because it was repeatedly asked every like lot of years I've seen. Now these questions are also very famous, but you need to know what is the term and what it measures. Okay, if that much you know, it is enough. What is peroxide value? It measures the extent of oxidation, how if the fat is oxidized or not. For these, these are very important when you in a food safety point of view. For example, if you're going to a street food vendor, and if you are collecting you have seen, if you have noticed, like vada sellers and all, they will have their, their their oil will be black in color because they keep reusing it. Now, when you keep on reusing it, what happens is it gets a lot of oxidation takes place, right? So how do you know what is the extent of this oxidation? How bad is that oil? So you'll just go to a lab and do this peroxide test, and you will get the peroxide value, which measures the extent of oxidation. You know how much of the oxidation has taken place in these fats and oils. Now, next is free fatty acid content or FFA content, which quantifies the amount of free fatty acids in a fat or an oil sample. Now, the next is saponification value. Saponification value is the determines the average molecular weight of all fatty acids in a sample. So it is used to assess the average chain length of the fatty acid. For example, if you want to know what type of fatty acids are present in a particular oil. For example, you got an unknown oil sample, and you do the saponification test. You will know the from the average chain length of the fatty acids, you can identify which fatty acid it is. Is it oleic acid? Is it comaric acid, etc. Now, coming to the next one, iodine value. It measures the amount of unsaturation or number of double bonds in the fats and oils. It is an indicator of the degree of unsaturation in a sample. Okay, that is iodine value. So this is one of the, in the beginning I have shown you a particular slide. The first slide I have shown you the different questions, right? One of the question is the degree of unsaturation in fat is estimated by which method? Now, I'm sure you can identify it as iodine value, because iodine value gives you the number of, how many of unsaturation or the double bonds, how many double bonds are there in the oils. It is given by iodine value. So you see, these are the different methods that we have learned and discussed. So these are different types of proteins, tests for fats, oils, etc. Now, coming to another very common question in the same pattern. Okay, match the following. Is the type of pigments in foods. These two type of questions only I have dealt with today. There is one is the test for carbohydrates, proteins, fats, oils, etc. Next is the type of pigments. Now, this also you need to know these names of these pigments and where is it found. Okay. So, first pigment is chlorophyll, very famous. We are all familiar with chlorophyll. I'm sure all of you know it also. Chlorophyll, green color. Where is it found? Spinach, all green leafy vegetables. All green leafy vegetables. So first is a chlorophyll, all spinach, kale, broccoli, green beans, parsley, green apples, whatever it is, whatever green things all of them contain chlorophyll. Next comes carotenoids. This is also something which is very common, or it's very familiar to all of you. These are all, the orangish, yellowish shades. Okay, carotenoids are yellowish, orange shades. Oh, so what are the main sources? Carrots, sweet potato, pumpkin, tomato, watermelon, papaya, all these contain carotenoids. Then comes anthocyanin. This is around purplish kind of color. So purple type of color, purple, blue, purple, that kind of color. Red grapes, blueberries, blackberries, cherry, red cabbage, red grapes. So violet shades. All those are anthocyanin pigments. Now coming to betalain. Betalain is red. Some varieties of radish, beetroot. So when you hear betalain itself, you will know, red colored. And there are a lot of other, like other kind of fruits or vegetables which contains leaves, which contains betalains, which are similar in color with beetroots, like Malabar spinach, all this contains betalains because they are all reddish purple by that kind of color of beetroots. Next comes flavonoids. Flavonoids is a yellow color. Okay, they are yellow plant pigments seen mostly in lemon, citrus fruits, apples, berries, and also found in dark chocolates, etc. So these are usually yellowish shades. Okay. Then comes turmeric, curcumin. Curcumin is a pigment which is found in turmeric and it's very famous and I'm sure all of you must be knowing. Then comes catechins, mainly present in tea, green tea, black tea, red wines, etc. So please make note of this. I'll be sharing the slide, but not just the slide. I want you all to write them down also. Please mention chlorophyll, what are the sources, etc. So that you will learn it very well. Okay. Then comes lycopene, mainly present in watermelon, grapefruit, guava, tomato, betanin, again, red beets. Then there's a color, annatto, usually used as a food color, for example, in butter. And for that yellowish, that pale color, you use annatto. Okay. Then comes paprika, gives you that red pepper, that capsicum, that color. Then cryptoxanthin, just present in oranges, papaya, peach, apricot. Lutein is there in dark, leafy greens and corn, egg yolks, etc. Then anthraquinones are seen in aloe vera. So these are the different pigments, etc. So we have discussed two type of questions which are very commonly seen. One is the type of testing seen in like all the nutrient tests, and there is and also the type of pigments which are commonly found in foods. Hope it is clear to all.